Thanks everyone for coming to this M2D2 reading group. This week we have Susana from Baker Lab at the University of Washington. She's a fourth-year PG student and she specializes in designing protein binders for bioactive peptides. And we're going to hear about that today. So, Mike is yours.
Yeah. Okay. Thank you. Thank you for the introduction. And yes, today I'm gonna discuss our recently published paper that actually focuses on designing the novel proteins to target bioactive helical peptides. And before I start with the presentation, I would just to give a big shout out to all of the co-authors on the paper, which are shown here because this was a highly collaborative effort. So to start, I would also like to explain why binding peptides could be challenging from a design perspective. So peptides are small chains of amino acids and tend to be very flexible or even not structured in isolation. They also provide very small surfaces areas to make interactions with. So previously, protein design has shown to be successful at achieving high affinity binding to large protein targets, which are very rigid and very concave, but peptides don't really meet these characteristics. And these peptides sometimes have a lot of exposed hydrophobic surfaces that can make the binding more challenging because these peptides could aggregate or bind to each other. So binding peptides has been going on for some years in the Baker Lab. So for instance, Keija in our group, she recently published a paper where she designed some modular binding proteins to polyproline to rich peptides that are not very well structured. And also Danny, he developed some protein binders that target beta-strand peptides, specifically some peptides that are involved in neurodegenerative diseases such as Alzheimer's disease. And something that these two methods have in common is that these binders make interactions with these targets through the peptide backbone. So basically, the protein scaffold has like side chains that interact with the peptide backbone. But in this study, we focus on helical peptide targets, and this approach cannot work very well here because helical peptides have like a very extensive interactions inside their backbones. So they present a different kind of challenge from the design perspective. And also it is well known that many of these helical peptides exert very important biological functions. So for instance, a B class helical peptides bind to class B GPCR receptors and exert like a wide range of biological functions. So here I'm just showing you a representation or a schematic of the parathyroid hormone that is partially unstructured in solution, and once it's bound to its receptor, it adopts a fully helical structure. So we targeted different sets of helical peptides. So here I'm showing you the parathyroid hormone, glucagon, neuropeptide Y, which are also widely used for diagnostic applications. So for instance, for diagnosing parathyroid disorders such as hyperparathyroidism, glucagon can also be monitored to evaluate the progression of diabetes, and neuropeptide Y is also used in the clinic to detect Alzheimer's disease. We also think that our designed proteins could offer some advantages compared to antipeptide antibodies because they have some properties that are very desirable for diagnostic applications. So for instance, it's known that the novel proteins are highly thermostable, so that's a very good advantage. They also provide very reproducible systems. So currently in clinic, a lot of people use polyclonal antibodies that display batch-to-batch variations, and even they can detect different epitopes every time. And so this could be problematic for achieving like a good diagnostic. And specificity is also very important for detecting these peptides. Many of these are synthesized as prohormones that achieve maturation through like cleavage processes, so they can lead to different peptides with very high sequence similarity. But with computational methods, we can overcome this problem, specifying or targeting a very specific amino acid sequence. The novel proteins can also be very cheaply produced with high yields and low cost in escherichia. And they provide highly tunable systems for diagnostics, as we can adapt our de novo proteins to existing biosensors, as I'm gonna discuss later in this presentation. So this project was very interesting because in the span of like a very short amount of time, many methods, especially like deep learning methods, starting like appearing. But when we first started this project, we used like old design methods, such as parametric design. So this strategy basically came out like we reasoned that if we design protein scaffolds with an open groove, that could be like a general solution for the helical peptide binding problem, because that would provide a shape complementarity to this helical peptide target. So I'm showing you like this here. So it's like something like a hot dog, right? So if you have like an open slot for a helical peptide, that will provide like very nice number of interactions that could achieve high specificity and high affinity at the same time. So we basically assemble a library of protein scaffolds by sampling the Crick equation, the Crick parametric equations. So we sample different parameters such as supercoiling and helix distance. So we generated helix bundles. So we had like 5 helixes in these scaffolds. 3 of them were making the groove, and there were 2 buttressing helixes at the back. And by sampling these parameters, we were able to sample different groove shapes in these scaffolds, as I'm showing you here in this schematic. And it's very nice to sample different group shapes, because that could lead potentially to different binding modes to different peptides. So at that time, we used RFGen and RFdoc to perform the binder design. And then we use a split luciferase assay to measure like affinity binding of some of these designs. So here I'm just showing you in the left the AlphaFold model of one of these binders against PTH, which is shown in purple. And on the right, I'm showing you a split luciferase assay that enable us to estimate a KD for this interaction. However, you can see that the binding interactions that we could achieve with this method was not very high, because this binder was calculated to have a KD of higher of 1 micromolar. So for actual applications, we need binders which are very tight, because these hormones are present in very low concentrations in blood. So this was promising, but not very useful. So luckily at the time, a new method came out in the Baker lab, or was developed, which is Rosetta Fold Inpainting, which is a method that enables to design sequence and structure at the same time. So this method, I mean, is based on previous methods like missing information recovery problem. So when we look at our parametrically designed binders, we notice that some of these designs were not fully containing the peptide. So we reasoned that if we could extend their binding interfaces, we could achieve higher affinity, just because we have like a higher number of interactions with the target. So we specified to the model like which helixes we wanted to extend. So we extended actually all of the helixes in our starting scaffold at random lengths. We explored different kind of lengths. Then we use ProteinMPNN to redesign the protein interfaces, and we use AlphaFold to predict our designs. And it was very nice to see that after this process, we were able to improve a lot the binding affinity of our weak binders. So for instance, straight out from the computer, we were able to increase the binding affinity from 1 micromolar or higher than 1 micromolar to 6 nanomolar through this process. Additionally, binder was very specific, because it didn't bind to a similar peptide, which is PTH-related peptide, which actually binds to the same GPCR receptor in nature. We tried this strategy as well with other targets, such as glucagon and neuropeptide Y, also by extending the interfaces of parametrically designed proteins, and we were able to identify a glucagon binder with an affinity of around 200 nanomolar, and for neuropeptide Y, an affinity of 3.5 micromolar. Additionally, we tested a different approach that also require the pre-specification of a scaffold. So in this case, we use DHRs, which are designed helical that also are like pretty much helical, and they display an open groove. So in this case, we took this library of scaffold, and then we threaded a ideal helix into this groove, and then we basically replace the sequence of that ideal helix with the sequence of our target peptides, and then we use ProteinMPNN to redesign the interface between the peptide and our scaffold. And then we use AlphaFold to filter our designs and test them in the lab. And in this case, I'm showing you some results for the secretin binder, which was we displayed an affinity of around 4 nanomolar. And also displayed, I mean, it was not very specific, it bound to a similar affinity to glucagon. So this was leading to like very high specificity. And we additionally tested this design approach by using different clinically relevant peptides such as GIP and GLP-1. And as you can see, we were able to also achieve very decent binding affinities through this method. So all of the methods that I just discussed require the pre-specification of a scaffold, the structure, or also the structure of your target. However, during the time, also in the lab, there was this, which is AlphaFold hallucination protocol that doesn't require to provide the structure of a binder of a target to make designs. So this previously used to design symmetric oligomers. So in this case, what we did, it's very nicely because you only need to provide the primary sequence of amino acids that you want to target. In this case, the amino acid sequence of our peptide targets. And you need to pre-specify like what's the length of the binder that you want to build. So for instance, we sample different binder lengths from 50, 60, 80 to up to 100 residues. So you provide this to a network. AlphaFold is able to predict the structures of the peptide in the context of the binder. So something important is that this method enables for flexible protein backbone design. And then the network is actually able to sequence search guided by Monte Carlo, and it's also guided by a loss function that takes into consideration some parameters that we know that correlate very well with binding. So for instance, PLDGT is a metric of like how good is your prediction. Interaction PAE tells you like how feasible like is this interaction, or like if it's highly probable that your binder is binding to your peptide target, and the number of contacts. So more contacts mean maybe higher affinity. And this is an iterative process until we converge into some backbones that we further design with ProteinMPNN. We again use AlphaFold to predict these designs. And in this, instead of focusing on hormone targets, we used BH3 apoptosis peptide target. In this case, we used BIF. And this is a very nice test case for this approach because this peptide is known to be unfolded in the absence of a binding partner. However, when it binds to its native target, protein, it falls into a helical structure. So here I'm just showing you some examples of some outputs of this method, and you can see that the peptide binds to its target in different conformations, and that's actually very nicely and could be very useful for some intrinsically disordered peptides. And again, after this process, we were able to identify very good binding hits. The best one displayed a KD of around 7 nanomolar, which is actually a tighter interaction compared to the native interaction of this peptide to its MCL1, a native target. And the trajectories of this process convert in around 5,000 steps. So again, like methods were coming out very quickly. So in the Baker lab, this diffusion method came out. I don't know if you're familiar with this, but this is a generative model which can build a protein structure starting from a random noise distribution. So in this case, a Gaussian noise distribution where you have like a cloud of atoms around a target, and then the model it starts decreasing the noise, until you have like some like protein look structure that falls around your specified target. So for this peptide application, the only thing that we provided was the structure of our peptide target together with some hotspot residues, some hydrophobic hotspot residues, and then we run diffusion. Here I'm showing you actually some movies, so you can see that you start with this cloud of atoms that are not very well ordered, and then they converge until they evolve around our helical peptide target. Something very interesting or very nice was to see that this method is super like binders straight out of the computer. For instance, our parathyroid hormone binder was binding with like picomolar-affinity to its target and was also very specific as it didn't cross-react with the PTH-related peptide. And this didn't require any further optimization or something. This was obtained after screening 96 designs. And additionally, we performed the same protocol using another BH3 peptide, which was BEAM. And again, we observed a picomolar affinity for this target. We were very lucky to solve the crystallographic structure of our beam binder. Here I'm showing the peptide in red and the binder in teal, and I'm just showing like some zoom-in views into the binding interface. And for instance, the crystallographic structure was able to recapitulate like this king helix that was predicted by AlphaFold. And also you can see that our binder is making some electrostatic interactions with the peptide, as I'm showing you here. So that was very, very nice. And actually want to say at the point that we noticed that using diffusion for this task was much more computationally efficient and better than the previous methods. So for instance, hallucination that only requires to provide the sequence of your peptide, we can also perform the same task with diffusion as Joe Watson. He fine-tuned the model so it can perform the same task. So we tested this by using peptide YY, which I'm showing you here in brown. So this peptide YY belongs to the same family as neuropeptide Y and also mediates very important biological functions. So here I'm showing you on the left the peptide bound to its GPCR receptor. So you can see that there are some regions in the peptide shown in teal that are unstructured. And on the right, I'm showing you the AlphaFold prediction of our best binding hit, where you can see that these regions that seem to be unstructured in the crystallographic structure adopt a helical structure in the presence of our binder. And also our binder is able to interact with very decent affinity to this target with around 24 nanomolar affinity and also high specificity. So besides this task, we were excited to test if the diffusion method was able to improve the binding affinity of weaker hits. So by sampling around the backbones of like initial designs. So in this schematic, I'm showing you on the left like this original design. So this is our neuropeptide Y binder, a weak hit. And you can see that in this partial diffusion, it involves two processes. You need to noise the structure and you decide how much noise you want to introduce to the structure, and then you need to denoise it back. So for instance, you have an original design, you introduce some amount of noise that you can control and you can sample different amount of noises, and then you denoise it back. So you end up having a structure that is similar to your starting structure, but it's not exactly the same. So for instance, I don't know if you can see here, but there's like some gray structure here, and that's basically the overlay of the starting design with our partially diffused design. And you can see that the backbones are a little bit shifted. And if we look at some metrics that correlate with binding affinity of the binding quality of our designs, such as the contact molecular surface, Rosetta DDG, and the shape complementarity interface, before the partial diffusion and after the partial diffusion process, you can see that all of these metrics actually improve significantly for this design. So we were very excited to see that these metrics were like improving, but we went to the lab and we actually test This and here on the left were original designs that we obtained by Inpainting that I showed you previously. And after running this partial diffusion approach, we were able to significantly improve their binding affinities. So for instance, for neuropeptide Y, we were able to improve the affinity from 3.5 micromolar to 5 nanomolar. And for glucagon, we were able to improve the binding affinity from 200 nanomolar to also picomolar-affinity, which was like super great to see because, again, you don't need to screen like thousands of design or make very intensive campaigns to improve the binding affinity of your starting designs. We were also very lucky to solve the crystallographic structures of the glucagon binder before the partial diffusion process and after the partial diffusion process. And I mean, that's very important to gain some insight of the structural changes that lead to this increase in affinity. So here on the top, I'm just showing in teal and yellow our partially diffused glucagon binder overlaid in gray with our Inpainted design. And you can see that they are very similar, like the structures are very similar, but there's an obvious backbone shift. And I showed you previously that the KDs of this binder significantly improve after the partial diffusion. So if we look closely, like what changed? So here I'm just showing you the crystallographic structure of the partially diffused. And if we look at the packing, so for instance, this overlaid green structure corresponds to the Inpainted version of the protein. So you can see that at this position, the Inpainted design used to have a phenylalanine that is like very bulky and is not necessarily very well packed in this cavity, but our partial diffusion is bulky residue has an isoleucine that is better packed in this condition. So this subtle backbone shift was able to accommodate better some residues. And also, if we look at this other position in our partially diffused design, we can see that our binder used to have a serine at this position, but after the partial diffusion process, there was this tyrosine which was positioned here, and this tyrosine is actually making an hydrogen bond with a peptide target, which could be beneficial for increasing the binding affinity. So the conclusion is that these subtle backbone shifts allow for better packing, better shape complementarity, and also could lead to better better designs because we can accommodate better other residues that can make extensive interactions with our peptide. I already said this, but if we compare the metrics, for example, in top for the glucagon binder and on the bottom for the neuropeptide binder in green, we can see that our starting designs that are shown in red and our partial diffuse that I'm shown in these black dashed lines, in almost all the cases, the metrics improve after the partial diffusion process. And this is actually this is broadly applicable. So we tested this for peptide binding, but currently in the Baker lab, we use this partial diffusion approach to for affinity maturation of basically any PPI kind of interaction. So this is like super useful for other targets that are not helical peptides as well. So yeah, just to conclude with this story, we were able to generate very high affinity peptide binders, and I told you at the beginning of my presentation that these peptides exert very important biological functions and are biomarkers. So we wanted to basically use them as diagnostic tools. So something that we first tried was to fuse our PTH Inpainted binder, the one that had a KD of around 7 nanomolar, to this Luke cage system. I don't know if you're familiar with this system, but this is a modular system where you have a cage and you have a key. So in this cage, you have a latch where you actually insert any binder of interest, and you have a split luciferase. Also, the key has the other half of the split luciferase. So in the presence of an analyte, in this case, our peptide, this leads to the opening of the cage, and then it exposes this split luciferase. Now the key can contact this cage, reconstitute the luciferase, so that leads to a luminescent signals. So we basically use this system for detecting PTH, and we measure luminescence over a range of PTH concentrations, and we use a linear regression to estimate the limit of detection of this biosensor. And it was very nice to see that the limit of detection of this biosensor matched well with the KD of this binder, which I told you is around 7 nanomolar. So in addition to this biosensor system, we collaborated with the Hoffenagel lab at the UW to implement our peptide binders as affinity reagents for mass spec detection. So these peptides are very low concentration in serum, so they normally require some enrichment for detection. So what we did in this experiment, which we didn't use any clinical samples, we used either plasma or buffer, which was spiked with our hormones of interest. And then we couple magnetic beads either with our mini binders, and for the glucagon binder, we also had a positive control with a monoclonal antibody. Then we incubated our beads with this sample. We performed some washes and sublimate and removal. In the case of the PTH sample, we performed some tryptic digestion, and then we used the mass spectrometry detection to evaluate if we could find these peptides in the sample. So here at the bottom graphs in purple, I'm just showing you the results for the PTH binder beads. And here on the x-axis are the different conditions that we use, and the y-axis, I'm showing you the recovery percent from these conditions. So here, the buffer plus PTH basically is just a positive control. So in this condition, you have ideally 100% recovery. If you have a negative control mixed with this PTH hormone, you don't get a lot of enrichment. But if you use either buffer or plasma in the presence of our binder plus PTH, you are able to like close to 50% recovery, which is very exciting to see. And for a glucagon binder, we were able to compare directly with monoclonal antibody against glucagon. So here we used, again, negative control. We don't see a lot of recovery. If we use a buffer, we can see that our mini binder performs very similarly to the monoclonal antibody beads. And something that we achieved with our mini binders that the antibody beads didn't achieve was to be reused. So we basically reused the same beads for another pull-down experiment. And we can see that our mini binder actually is able to recover a significant amount of peptide after this second pull-down experiment. But the monoclonal antibody is not able to recover any of this peptide any longer. And this is presumably due to the high stability of our mini binders. Our mini binders are, I didn't show it here, but they can be basically boiled up to 95 degrees and they're still folded. And antibodies, of course, they are not as stable. So probably this is behind the why this is not working with a monoclonal antibody. So after everything that I told you, I will like to give you like some points. What did we learn through this study? So we learned that we can use different sets of computational approaches to design high affinity binders for helical peptides. But interestingly, all of them converge into the same solution, which is like a group shape scaffold, right? It doesn't matter if you use like hallucination or if you use diffusion, the models always end up building something that has a groove. And we also, when we started the project, we ambitioned the same solution, which is very interesting. We also show that diffusion, it's like very powerful technique to design peptide binders either by scratch or by partial diffusion to improve the of starting designs. I already also mentioned that this in silico affinity maturation is able to improve the binding affinity of these peptide binders, but should be broadly applicable. So it could improve the binding affinity of almost any binder to any other peptide or protein binder. And we also demonstrate that these designed binders could be used as affinity reagents for diagnostic applications. And I mean, for like real applications in like with real samples, more studies needs to be done. But I think this is a very exciting start for that. And with that, I would just like to thank you to all of the people who were involved in this project. And yeah, I'm ready to take any questions. Thank you.
Great. Thanks for your talk. If there's any question in the audience, feel free to raise your hand, be on mute. In the time being, I can ask the first one.
Yeah.
So I'm wondering if for the partial diffusion setting, does it make sense to like fix certain parts that you want to like not change or are very important, or did you just let it be free and kind of
Yeah,
redesign stuff?
that's a very good question. Actually, it is possible to fix some parts for the partial diffusion. It's actually like explained on the GitHub of how to do that. I mean, some people have explored partial diffusion, for example, for a small molecule binding, but you can imagine that binding like a large peptide or a small molecule are very different challenge, because for binding small molecules, you need to have like high accuracy on the interaction, so you don't want to disturb everything, so you could potentially fix some residues. But for this project, we actually partially diffused everything. But yes, it's possible to fix some regions before the partial diffusion. It depends on the application.
Yeah, that makes sense. Any audience questions? Yeah,
I had a
Sean,
question.
go ahead.
Yeah.
Oh,
Sorry, go ahead, Sean.
Oh, it was kind of related to the previous question, actually. Thanks for the nice talk, by the way. You mentioned that there are hotspot residues that kind of stayed fixed during that. I guess, how were the hotspots decided upon? And then B, I guess, were you able to like quantify or like determine how much those contributed, like holding that fixed contributed to your end result?
Yeah.
Yeah, that's it.
Yeah, that's a very good question. So for the unconditional binder design, where we only provide the structure of our target, like kind of like the standard way to choosing hotspot residues are normally choosing some hydrophobic residues. So normally you provide like the maximum 5 hotspot residues. It can be 3 to 5 hydrophobic residues. And I think it works pretty well, at least for helical peptides, which are amphipathic. I mean, also depends, there are some peptides that are very charged and could be more challenging to bind. But I think just hydrophobic residues are very useful. And I don't think we have quantified, or at least I don't know, like the answer to your second question of how that much matters for the end result. But yeah. I think there was another question.
yeah, Griffin.
Yeah, my question was about doing like the reverse. So if you have an existing protein that you want to design a peptide to bind to, can you enforce like structural constraints on that peptide with the diffusion process? Like if you wanted to design only like helical peptides or cyclic peptides?
Yeah, I think, yeah, that's I mean, some people in the lab are actually interested on that. I haven't really explored that, but that's definitely possible, and there's people in the group who are currently working on that.
Okay, thanks.
I have the question. Did you end up doing any like multi-objective optimization, or was it mostly like generate and filter kind of procedure?
Sorry, for what?
I guess for most of these experiments, like there's multiple things you might care about, or is there just one signal that you can optimize and it works out?
For the filtering?
Yeah.
I mean, yeah, for filtering, I mean, honestly, I think the most important filters that we use right now for protein interaction design in the Baker lab are PAE, like at the end, like after AlphaFold prediction. I think PAE is the best metric that we have found that correlates with binding. So if PAE value are below 10, it will mean that you have probably like a very good binder. We also use PLDTT, and I mean, sometimes I use DDG. So I think those are the main filters that we use. I think for this contact, the shape complementarity or surface contact surface could also very good indication of the quality of your binder, but I think almost PA and PLDT must be very good for filtering.
All right. Any last questions from the audience?
I had a question. So it seemed like in the results that you presented, like you got a low nanomolar binder with like the collab design approach, and then like a nanomolar or a picomolar binder with their diffusion approach. Do you think it's just like a strictly better method, or are there still like applications for the older method?
For which peptide? Sorry, like you mean for the hallucination or which one?
I don't remember the specific one, but you had one where you had like a picomolar binder.
I mean, yeah, I think the picomolar binders were obtained with diffusion, the diffusion methodologies. Yeah.
Right, yeah.
Yeah.
So do you think it's like still worth running the other like protocols since there's like such a large difference in?
Yeah, I think honestly, we explored all of these methodologies because they were coming, like out very quickly. But I think like if I was gonna start a new project of like peptide binding, I would only try diffusion. Because it's actually the hallucination protocol that I explained that leads to good binding. Actually, it's not very computationally efficient. Like diffusion can make the same much more faster. And in painting, for instance, no one like use it anymore. So I think, yeah, diffusion is the way to go. And I think, for achieving picomolar affinity binders, I mean, if your target is kind of easy as the parathyroid hormone that I show, it's like completely doable. But in other cases where you have like a more flexible and structured peptide, you will require like a partial diffusion approach just to sample around your initial target to get very good binding affinities. And yeah, I mean, the other methods I'm just showing because it's like an historical like overview of the methods, but I think diffusion is the way to go. Yeah.